Hello everyone, I'm Giovanna Ordonez, a fourth year MIMG Pathway 2 major. I do research at Boyer Hall with Eric J. Dietz. This is a mostly computational biology lab, however, my focus has been in vitro experiments to understand how the bacterial proteasome core particle is assembled. So, what is the proteasome? The proteasome is a macromolecule complex designed to control the degradation of abnormal or short-lived proteins in an organism as part of its cell survival. This barrel-like structure is composed of a heptameric alpha in green and heptameric beta in blue subunits. Degradation occurs in the middle beta region. However, this catalytic portion is protected by propeptides during the assembly process. The proteasome is an essential part of the cell cycle. This is important to a lot of the cell's function naturally associated with disease. So why is it so important to us? Conserved architecture for this core particle is seen in all forms of life. So we see this in archaea cells, bacteria cells, and eukaryotic cells. Furthermore, these cells cannot transcribe an active proteasome core particle, meaning without alpha and beta subunits assembling the core particle, there is no active proteasome that can control the degradation of proteins. So if we understand how the proteasome is assembled, we can target it for new assembly inhibitor therapies. This has never been done before with Mycobacterium tuberculosis, also known as MTB, which MTB proteasome core particle assembly inhibition has been a topic of discussion for treating tuberculosis. This is a new way of approaching how this assembly process occurs. So let's look at this broadly accepted pathway in literature for the proteasome. In archaea, on the top, we see that it uses a heptameric alpha ring as a template for beta to bind, creating the half proteasome. So this ring is used as a template where beta comes and joins like little Legos, creating the half proteasome. Two half proteasomes are formed, the propeptide sleeve, and the core particle is created. In bacteria, this uses an alpha and beta dimers to come together and build the half proteasome. So two half proteasomes then come together, propeptide sleeve, leaving an active core particle. However, there's an unordered pathway proposed by published work done using Rhodococcus, suggesting that an unordered pathway where alpha and beta dimers are first formed and any combination of alpha and beta binding creates the haploidosome, where both haploidosomes come together, propeptide sleeve, and we get our active core particle. So we believe that MTB proteasome core particle assembles in an unordered formation of the half proteasome following a separation of timescales between half proteasome formation and dimerization to form the core particle. In the case of MTB proteasome core particle assembly, we don't know if it follows bacteria assembly accepted widely in literature or an unordered formation seen in Rhodococcus. We are essentially asking, how does the half proteasome know how to dimerize, but not dimerize with near intermediates? Basically, how does it know how to assemble to create this core particle? However, in vitro experiments and purification of the MTB proteasome core particle subunits have been difficult to replicate, despite novel methods developed by Anupama Kante, a former lab member at Eric J. Dietz' lab. And to have experiments, you need the protein. This is just some of the data that I've acquired in the last couple of months testing various sonication conditions. This is just one example of our newly established purification for alpha. For day 1 purification, we use immobilized metal affinity chromatography, where we expect to see our construct below. It is a N-terminus with a his tag, TEV, and our protein of interest, alpha. We expect to see our construct below in our third peak. This fraction peak is then collected to be dialyzed and purified again the following day. We also analyze our protein through an SDS page that you can see on the right. So for day 2 of purification, we expect to see our purified protein in the flow through. So once we dialyze, this his and TEV would theoretically cleave off. So we would see this in our flow through. since the his tag and TEV and other residues will bind to the his trap that we use during our immobilized metal affinity chromatography. This has been a huge achievement since the last successful alpha purification was more than 2 years ago. We are hopeful that through this steps of optimization and figuring out how to purify it more successfully, we can go through further experiments such as running various assembly assays and assembly intermediates to characterize the size and structure through cryo-EM and NMR. Thank you so much. I would like to acknowledge my lab members here and my cat, of course. And here is just our little lab photo. Thank you so much for your time.